in our last session. If you remember, we first discussed the structure of the urinary system and the organs that will share in the formation of the urinary system. Those are kidneys, the ureters, urinary bladder, and the urethra. We discussed the structure of the kidney. Remember, when we have a sagittal section of the kidney was formed of two layers. An outer layer, this was my renal cortex. My inner layer, this was my renal medulla. If you remember, at the junction between the renal cortex and renal medulla, we did have those pyramidal shaped structures. Those are what we call the renal pyramids. And remember, the tip of the pyramid where we're going to be accumulating the urine to be released in the network of ducts. This was the renal papilla, or the papilla of the pyramid. The urine will then be conducted to small ducts. Those are what we call the minor calyces. And those minor calyces are going to be conducting the urine into larger ducts. Those large ducts are what we call the major calyces. And the major calyces will then conduct the urine to a funnel-shaped tube. This is what we call the renal pelvis. The renal pelvis will allow the urine to flow to the vascular tubes that will conduct the urine down to the urinary bladder, and this vascular tube on here is what we call the ureter. If you remember, we stated that the functional and structural unit of the kidney was the nephron, and the nephrons are formed of tubules, and running parallel to them, we've got our capillaries. So nephrons, we have 1 million of them per kidney. They are formed of those structural units on here. Those are my nephrons, and again, nephrons are going to be formed by kidney tubules, as you see on here, and running parallel to them, we've got tufts of capillaries. First part of the kidney tubule was a cup-shaped tube. This is my Bowman's capsule. The set of tubules that would be close to the Bowman's capsule, this was my proximal convoluted tubules. Then I will pass through the loop of Henle. Loop of Henle, and then the distal convoluted tubules, and finally collecting the urine from the different nephrons are going to be my collecting ducts. The associated capillaries that will be sharing in the formation of the nephrons. First tuft of capillaries that we've seen, those were located inside the Bowman's capsule. and we call those are my glomerular capillaries, which are forming my glomerulus. Remember, the glomerulus has a large afferent arteriole that brings much larger amounts of blood than the arterioles that drains it, which is my efferent arteriole. And this is what allowed the glomerulus to have a high hydrostatic pressure, which, as we're going to see in a minute, will allow the filtration to take place. Remember, as a continuation of the efferent arteriole, I've got tufts of capillaries that will be surrounding my proximal and distal convoluted tubules. Those are what we call the peritubular capillaries. Peri means around, and they are located around the tubules, so we're going to call them my peritubular capillaries. The third type of capillaries that we've got was a set of vessels that will run straight surrounding the loops of Henle, and they are prominent in the nephrons with long loops of Henle. Those are vessels that are running straight. This is my vasa recta. Remember, we did have two types of nephrons. We've got cortical nephrons, which are going to be located mainly in the cortex of the kidney. And we got juxtamedullary nephrons, and the juxtamedullary nephrons, as you see on here, they are still located in the cortex, but at the junction with the medulla. And they will be having long loops of Henle surrounded by vasa recta. So the vasa recta on here are the straight vessels that are surrounding the long loops of Henle. If you remember, we discussed last class the structure of the filtration membrane, which is the connection between the glomerular capillaries and the Bowman's capsule. If you remember, the glomerular capillaries are fenestrated capillaries. So the first layer of the filtration membrane is formed by fenestrated endothelial cells. Those are the cells of my glomerular capillary. On the other side, I have got the cells of the Bowman's capsule, and those are cells with food processes. Those are what we call the podocytes. Remember, both the fenestrated endothelial cells and the podocytes are going to be sitting on a gel-like basement membrane, which is formed by the lamina of the two sides. This gel-like basement membrane was negatively charged. Remember, anything can travel from the glomerulus to the Bowman's capsule except for three main things. Those are, first, the blood cells. Blood cells are larger than the fenestrations of the endothelial cells. so they cannot travel through the fenestrated endothelium to get filtered. So it's not something normal to see blood cells out in the filtrate or in the urine. Other things that we don't see getting filtered where the anions or mostly the negatively charged plasma proteins. You remember, plasma proteins are negatively charged, the same like my gel-like basement membrane. So I've got repulsion between the two. So plasma proteins will fail to get filtered through the filtration membrane. Third thing that won't be able to get filtered were the macromolecules. Remember, macromolecules are large structures. They won't be able to travel because they are larger than the filtration slits between the feet of the podocytes. So three main things that won't get filtered. This means for me that filtrate is equal to the fluid around the blood cells, which is the plasma, but without proteins. So filtrate is equal to plasma minus the plasma proteins. If you remember, each drop of the plasma gets filtered how many times? Do you remember? How many times do I filter the plasma per day? How many times do you filter the plasma per day? 60 times. So I'm filtering large amounts of fluid. But all the filtrates that I'm releasing out in the Bowman's capsule is not by any means closer to what I release out of the body as urine. Urine is representing less than 1% of the total amount of fluids that gets filtered. So the GFR or the glomerular filtration rate is the volume of the filtrate that gets formed per minute. And this is going to be around 120 to 25 milliliters per minute. So what's going to be driving the fluid to get filtered? So we know that everything can get filtered except for plasma proteins, blood cells, and macromolecules. But what's actually going to be driving this fluid to exit the blood vessels and to travel towards my Bowman's capsules, to move from the blood to the Bowman's capsule for it to be the filtrate? I have important pressures that play a role in here. And according to them, we're going to be able to allow the filtration to take place. So if we're looking on here at the blood vessel, this is again my glomerular capillary. And on the other side, this is going to be my Bowman's capsule. First, I have the pressure of the blood flow going to the inside of those glomerular capillary. And this is going to be determined by the tissue perfusion. The amount of fluid that I get in creates a pushing force that will be driving the fluid to move from the blood to the outside through the fenestrations of the endothelial cells. Remember, what is the blood pressure? It's the pressure that will be exerted on the wall of the vessel. So here, if I'm exerting pressure on the vessel, which is porous, so it's going to be leaking fluid to the outside. What do we call this pressure? It's my glomerular hydrostatic pressure. It's going to be pushing fluid from the vessel to the Bowman's capsule for this fluid to be filtered. Do I have any fluid on the other side? Do I have any fluid in the Bowman's capsule on here? What do you think? Do you have any fluid in the Bowman's capsule? What do you think? Do you have any fluid in the Bowman's capsule? Yes. What is the name of the fluid that you have in the Bowman's capsule? Can you remind me? The one that was able to move from the blood to the Bowman's capsule. This is what we call, again, filtrate. So do I have fluid? Do I have fluid in the Bowman's capsule on here? Yes, it's called filtrate. And this filtrate, is it something or nothing? Is it like nothing, or is it liquid, and this liquid has a weight and creates a hydrostatic force? What do you think? Is it something or nothing? Does it have a weight or not? It's liquid, right? So this filtrate also gonna be applying a hydrostatic pressure, a pushing force. So some of you will not be convinced. How come this fluid did just move out of the vessels? How it's going to be trying to move backwards? So I will remind you with this tube that we have been drawing at the end of last class. This U-shaped tube. So first scenario here that we're looking at. I added 1 liter of water on here, on this side, and I have nothing in the tube. And I've got another tube. And I will be adding also 1 liter, but I have fluid on the right side of the tube. Here I don't have fluid. Here I have fluid. Which one gonna be able to travel from this point to this point faster? The one that's gonna be faced by this water on the other side, or the one that doesn't have any water on the other side? What do you think? With no water. Why? Because this water on the right side tends to move in the opposite direction. So it's going to be creating a resistance. So this water, because of its weight, is trying to move in this direction. The water I'm adding is trying to move in the opposite direction. This is the same exact concept of the hydrostatic pressure on both sides. So in the blood, I have a hydrostatic pressure called the glomerular hydrostatic pressure. And in the Bowman's capsule, I have filtrate, which is fluid. And this fluid also going to be trying to move to the opposite side. So what do we call this fluid on here? This is called my capsular hydrostatic pressure. So again, what we're looking at in here, it's the hydrostatic pressure that will be created due to the presence of the fluid and the force by which the fluid is going to be traveling with. All right. Another important force that will be affecting the movement of the fluid is going to be due to the presence of solutes. If you remember, I have two tubes like this that are connected together, and I've got a membrane that is impermeable for the solute in between the two. One side has a high solute content, and other side did have a low solute content. And I did have the same volume on both sides. What's going to happen to the water? Will it move from right to left or from left to right? What do you think? Will I be moving from right to left, one, or left to right, two? Which one do you choose? Remember, what's by definition is osmosis. Osmosis is the movement of water from the side with low solute to the side with high solute. So water is going to be traveling from where to where? From right to left. Exactly. So what I will end up with, I will end up with the tube that is looking like this. With equal concentration of solutes. So the water did move from the side with low solute concentration to the side with high solute concentration until I equalize the solute concentration on both sides of the membrane. Is this clear? Is this clear? Osmosis? Is osmosis clear? All right, so which side has more solutes? What do you think? Your blood or the filtrate? Which side has more solutes? Blood one, filtrate two. Which one? One, why? Why the blood has more solutes? Because you didn't allow the blood cells to travel. You didn't allow plasma proteins to get filtered. Macromolecules didn't get filtered. So you have a lot of stuff that don't get filtered, they remain in the blood. So which side is gonna have a higher solute concentration is gonna be my blood. So the glomerulus here has higher solute concentration. So this means what for me? If I have higher solute concentration, would I be pulling water towards me or would I be pushing the water away from me? High solute concentration on here, lots of solutes. What happened to the side with high solute? Did it pull the water or push the water? have high solute. So am I pulling the water or push the water? What happened? You're pulling the water. I'm pulling the water towards me. So here, as I have much higher solute content because a lot of stuff didn't get filtered, what's going to happen? The water is going to be pulled backwards towards your blood vessel. What do we call this pressure? Is my blood colloid osmotic pressure. So putting this into consideration, looking at the effective pressures, would I have filtration or not? How many pressures did we see that will be enhancing or allowing the filtration to take place? How many are pushing the fluid from the capillaries, from the glomerulus, from the blood to the Bowman's capsule? How many? How many forces? Just one. And how many forces are getting the water to move from the Bowman's capsule back to the blood? What do you think? Two. What are those two? I have capsular hydrostatic pressure and I have the blood colloid osmotic pressure. Those are two pressures that will allow the movement of fluid back to the vessels. And the one that allows the filtration to take place is the glomerular hydrostatic pressure. So would I have any filtration? What do you think? Would I end up having any filtration? If I have one force that will be pushing the fluid out and two forces pulling the fluid in, what do you think? Would I have any filtration? If one is pushing and two are opposing, would I end up having any filtration? What do you think? I will end up having filtration because the hydrostatic pressure of the glomerulus is equal to 55 millimeter mercury. The capsular hydrostatic pressure is equal to 15 millimeter mercury. And the blood colloid osmotic pressure is equal to 30 millimeter mercury. So what is the pressure by which I'm pushing the fluid out? I'm pushing the fluid out with. It's 55. And what are the pressures that are opposing the filtration from taking place? 15 and 30. So what is the net filtration pressure? What is the effective pressure that I actually gonna be pushing the fluid with? 55 minus 30 and minus 15. This means that my net filtration pressure, the effective pressure that allows the fluid to exit, 55 minus 45 is going to be 10 millimeter mercury. All right. So again, what is the net filtration pressure? It's the difference between the pressures that will be pushing the fluid out of the vessel and the pressures that will be opposing the filtration. So hydrostatic pressure is greater then both the capsular hydrostatic pressure and the blood colloid osmotic pressure combined by 10 millimeter mercury. And this explains why it is so critical to maintain normal blood pressure at the kidney. Because if I do have a major drop in my hydrostatic pressure, so let's say my blood pressure did drop, and as a result, my tissue perfusion declined, causing a major reduction in my blood glomerular hydrostatic pressure. So rather than being 55, it drops to 45. So what this will result in? I won't have any filtration. So no filtration means no urine filtration, no urine formation. And if no urine is going to be formed, what's going to happen? I will be accumulating toxins in my system. And a major reduction of the filtration is going to result in renal failure. All right, so we all did agree that the kidney hates high blood pressure or low blood pressure. What do you think? What do you think? Does the kidney hate low blood pressure or high blood pressure? Low blood pressure, exactly. Low blood pressure. So it's going to be trying all the time to increase the blood pressure back to normal. All the time it's going to be increasing the blood pressure back to normal. This is the first part of your essay question for this upcoming lab exam. So in case of low blood pressure, how is the kidney going to respond? First, we need, in order to understand how the kidney responds, we need to study a set of corpuscles that are present in the kidney, one at each nephron, one per nephron. This is what we call the juxtaglomerular apparatus. Juxtaglomerular apparatus is going to be formed of 3 sets of cells. It's a cluster of cells that are present very close to the glomerulus, and this is where I got the name, juxtaglomerular. I am located close to the glomerulus. If we're looking on here, this is my Bowman's capsule, like this. This is my proximal convoluted tubules. Loop of Henle, descending limb, and here this is my ascending limb. So the juxtaglomerular apparatus is a cluster of cells that will be located close to the glomerulus. First set of cells that will be sharing in the formation of this cluster is going to be the modified smooth muscle cells of the afferent arteriole. And those modified smooth muscle cells going to contain cytoplasmic granules. And the content of those granules is a hormone that we call renin. And as those smooth muscle cells will be exhibiting cytoplasmic granules, we call them the granular cells. Granular cells are going to be responsible to detect changes in the blood pressure according to the degree of stretch applied to the wall of the afferent arteriole. According to the degree of stretch, whether those cells going to be excited, they will start to release renin or they wouldn't. Here, they are what we call mechanoreceptors. What do we mean by mechanoreceptors? Those are receptors detecting mechanical change. And here we mean by the mechanical change, the degree of stretch applied to the wall of the vessel. So again, the very first type of cells that will share in the formation of the juxtaglomerular apparatus, which is going to be responsible for the regulation of the blood pressure, are going to be the granular cells, and those are again modified smooth muscle cells that contain cytoplasmic granules containing renin. They are responsible to detect the degree of stretch to get an idea about the state of the blood pressure. The second type of cells, those are modified told cells which are closely packed. They are located in the ascending limb of the loop of Henle. So if we're looking closer on here, I have cells of the ascending limb of the loop of Henle. Those are closely packed together. And they are located very close to the granular cells. We call those cells are my macula densa. Those are going to be acting as chemoreceptors. What do I mean by chemoreceptors? They are detecting chemical change. Chemoreceptors have the ability to sense the concentration of sodium in the filtrate, and this gives you an idea about how well are you handling the regulation of the blood pressure. So again, we've got three types of cells forming the juxtaglomerular apparatus. The first set of cells, those are called my granular cells. Those are modified smooth muscles that contain cytoplasmic granules, and inside those cytoplasmic granules, you keep a hormone called renin. The second type of cells that will share in the formation of the juxtaglomerular apparatus is my macula densa, which are modified told cells of the ascending limb of the loop of Henle. They are acting as chemoreceptors to detect the sodium concentration in the filtrate. A third type of cells that will share in the formation of my juxtaglomerular apparatus would be a cluster of cells that will be found between the granular cells and the macula densa. Those cells will serve as a channel of communication between the granular cells and the macula densa. In order for them to allow the fastest possible communication to take place between those two types of cells, those cells will have gap junctions in between. And remember, gap junctions, those are the cell junctions that will be attaching the cells to one another, but in the same time allow the electrical signals to travel freely between the cells. We call this type of cells, those are my extraglomerular, because you see on here, I am located outside of the glomerulus. This is my glomerulus on here, located inside of the Bowman's capsule. So they are extraglomerular mesangial cells. Again, extraglomerular mesangial cells are interconnected with gap junctions and will allow the passage of signals between the macula densa and the granular cells. Questions? So again, kidney is gonna hate if you get exposed to a state of low blood pressure. So it's gonna be trying to increase your blood pressure whenever you have a drop in the blood pressure. So what part of the kidney exactly is gonna be responsible to regulate my blood pressure? It's gonna be the juxtaglomerular apparatus. And again, we've got three different types of cells that will form the juxtaglomerular apparatus, and those are my granular cells, my macula densa, and my extraglomerular mesangial cells. Those are the three types of cells that will build up the juxtaglomerular apparatus. Again, what are the granular cells? Those are modified smooth muscle cells of the afferent arteriole going towards the glomerulus. And those modified smooth muscle cells will act as mechanoreceptors. They are detecting the changes in the blood pressure by checking the degree of stretch applied to the wall of the arteriole. And they contain cytoplasmic organelles that will be the source for a hormone called renin. The other type of cells that we've got forming my juxtaglomerular apparatus is going to be my macula densa. Again, macula densa, those are modified told cells of the ascending limb of the loop of Henle, and those cells are going to be responsible to detect chemical change in the filtrate. So I'm checking here the amount of solute that is going to be released out in the urine. And accordingly, I will either decide to retain or not to regulate my blood pressure. Because remember, whenever you got sodium, water will follow by osmosis. So whenever I recapture the sodium, I retain water. And if I retain water, my blood pressure goes up. A third type of cells that share in the formation of the juxtaglomerular apparatus. Again, this is my extraglomerular mesangial cells, and they serve as a communication channel between the granular cells and the macula densa. We're looking closer on here. This is my afferent arteriole. The modified smooth muscle cells on here in the afferent arteriole, those are my granular cells. Modified told cells of the of the ascending limb of the loop of Henle. Those are my macula densa. And located in between the two, we've got those cells on here. Those are the extraglomerular mesangial cells. So what happens if I got a drop in my blood pressure? If I've got a decline in my blood pressure, this will reduce the amount of blood that would be delivered to the afferent arteriole. Can you remind me how many afferent arterioles do we have in the body? Around what number? What do you think? What do you think is the number of afferent arterioles in the body? Millions? Yeah, we've got 1 million per kidney. So you've got 1 million nephrons per kidney. And you have one afferent arteriole for each of those nephrons. So you've got 1 million afferent arterioles. So you've got 2 million afferent arterioles. Yes, exactly. So as the blood pressure drops, what happens is the amount of blood that will be reaching those afferent arterioles is going to decline. And this will definitely reduce the tissue perfusion to the kidney, reducing its hydrostatic pressure. So the granular cells will detect that there is a decline in the degree of stretch. Remember, if I don't have enough stretch to the wall of the afferent arteriole, the granular cells will detect this decline in the degree of stretch and will start to release the granular content, which is my renin. How's the renin going to act? So those granular cells going to be releasing the renin out in the blood. Renin doesn't increase the blood pressure directly. Instead, it's actually going to be activating a plasma protein called angiotensinogen, which is an inactive form of the hormone, into a more active form, but it's not fully active, called angiotensin I. Angiotensin I needs to become activated into angiotensin II, which is actually the effective hormone that increases your blood pressure. So if we look on here, what actually going to be activating angiotensin I into angiotensin II is an enzyme called the angiotensin converting enzyme, or ACE. If you're familiar with the different medications for hypertension, you will know that a family of medications that is very commonly used to treat hypertension is called ACE inhibitors. What are ACE inhibitors doing? They are inhibiting the activity of the angiotensin converting enzyme, preventing the transformation here of angiotensin I into angiotensin II, which is the active form of the enzyme. All right, so again, what we're looking at in here, we're looking at a decline in the blood pressure that resulted in a reduction in the degree of stretch of the afferent arterioles. This resulted in the activation of the granular cells that made them release renin. And again, how renin acts, they don't have a direct effect on the blood pressure. It acts by activating a plasma protein called angiotensin into a more active form, which is angiotensin 1. Angiotensin 1, it's not the final active form of the hormone. It needs to be activated into angiotensin 2 in order for it to be capable to increase the blood pressure. Again, what enzyme do I need in here in order for me to allow the conversion of angiotensin 1 into angiotensin 2? I need angiotensin converting enzyme, or ACE. And again, where is the angiotensin converting enzyme is going to be located? It's going to be located in the capillary endothelium. So can you remind me, how do we express here the blood pressure? What are the three main factors that will affect my mean arterial pressure? What are the three main factors that will be affecting your mean arterial pressure? Exactly, cardiac output. What else? Total peripheral resistance. And the third one is blood volume. Exactly, thank you. And can you remind me, what is cardiac output? What's cardiac output? What's cardiac output? It's the amount of blood that I pump per 1 minute. And how did we calculate the cardiac output? This is the amount of blood that you pump per beat times how many beats per minute, which is my heart rate. Stroke volume times heart rate. All right. So mean arterial pressure is going to be affected by all those factors. And if you remember, three factors that did affect the stroke volume, and so they will be affecting the mean arterial pressure. Those are my afterload, preload, and contractility. Those are the three main factors affecting my stroke volume. So you see, all those factors together are going to be regulating my mean arterial pressure. So how angiotensin II going to be acting in order to increase the blood pressure back to normal? Right. First action of the angiotensin II is by constricting the arterioles. I have vasoconstriction of the arterioles. And as I constrict the arterioles, what's going to happen to my total peripheral resistance? It's going to be going up. It's going to increase. So I have a rise in my total peripheral resistance. Rise in my total peripheral resistance going to result in what in terms of blood pressure? What do you think? When you increase total peripheral resistance, what's going to happen to your mean arterial pressure? It goes up, it increases. Another way by which angiotensin II is going to be increasing your mean arterial pressure is by enhancing the sodium reabsorption, either directly or indirectly. So angiotensin II gonna act on its own receptors located on the tubular cells of the nephron, and it will enhance them to reuptake the sodium from the filtrate back to the blood. And remember, when you reabsorb sodium, what's gonna follow the sodium? It's gonna be water. So what's gonna happen if I get water back to my blood vessels? What's gonna happen to my blood volume? What do you think? If an amount of water was supposed to be excreted out in the urine, but I instead reabsorb it back to my blood, what's gonna happen to my blood volume? What happens to the blood volume? It increases. And when you increase the blood volume, what's going to happen to your blood pressure as a result? Remember, the higher the blood volume, the greater the blood pressure. So as I reabsorb the sodium back from the filtrate back to my blood vessels, it's going to be followed by water. And as I reabsorb water back to my blood, this increases my blood volume, resulting in an increase in my mean arterial pressure. So this is the direct effect. The indirect effect is by stimulating the release of a hormone called aldosterone. If you remember, aldosterone is a hormone that gets released from the adrenal gland. To be more specific, the adrenal gland did have 3 layers on the outside, forming its cortex and an internal core, which is the adrenal medulla. Remember, those 3 layers are my zona glomerulosa, zona fasciculata, and zona reticularis. Aldosterone is a mineralocorticoid that gets released from the zona glomerulosa of the adrenal cortex. So what did stimulate here the zona glomerulosa to secrete the aldosterone? It's the angiotensin II. So angiotensin II itself is going to be enhancing the sodium reabsorption, and at the same time, it stimulates the release of aldosterone, which will also be enhancing the sodium reabsorption back to the blood vessels. So I have 2 ways by which I am stimulating the reabsorption of the sodium. I have a direct effect on the kidney tubules and an indirect effect by triggering the release of aldosterone from my adrenal cortex. And again, as I reabsorb sodium, water will follow, and as a result, I have an increase in the blood volume and so the blood pressure. Next, way by which the angiotensin II is going to be increasing the blood pressure, it's going to be by stimulating the brain. 2 parts of the brain that we're looking at here, both are located in the hypothalamus. I have the region in the hypothalamus, which is responsible for the thirst sensation. And recall, this is my hypothalamic thirst center. The other part of the hypothalamus that gets stimulated by the angiotensin II are going to be the nerve cells, which are responsible to release the hormone that will be stored and then released later on from the posterior pituitary gland. And we call this hormone ADH, or anti-diuretic hormone, or the other name of the ADH or anti-diuretic hormone is vasopressin. So how angiotensin II, by stimulating the hypothalamic thirst center and the release of ADH from the hypothalamus and the posterior pituitary gland, I'm going to be able to increase my blood pressure. So looking here, first at the hypothalamus, the hypothalamic thirst center. Once stimulated, what would you get as a feeling? I will have a thirst sensation. And when I have a thirst sensation, what would I do? I will drink a beverage. And when I drink, if I have a low blood volume, my blood volume gonna be increasing back to normal because of the absorption of the water in my intestines. And this will increase the blood volume and so my blood pressure. How ADH is going to be increasing my blood pressure? Remember, ADH stands for anti-diuretic hormone. Its main function is to insert channels, specific channels, in the collecting ducts and the distal convoluted tubules in order to allow the reuptake of the water back from the filtrate to my blood. This is the function of ADH. I am preventing. Diuresis means water loss in the urine. So the prevention of the water loss is taking place here under the effect of ADH by inserting aquaporine channels in the collecting ducts and the distal convoluted tubules, allowing more water reabsorption. So here I am retaining the water in the blood, and I'm not allowing this water to move out in the urine. So what's going to happen as a result to my blood volume? In this case, my blood volume goes up, and as my blood volume goes up, my mean arterial pressure goes up. Questions? Another way by which angiotensin II is going to be increasing my blood pressure is by constricting the efferent arteriole of the nephrons. So how, when I constrict the efferent arterioles, this is going to help me increase my blood pressure. If you remember, did I have a high blood pressure or a low blood pressure in my peritubular capillaries? Do you remember? Here, did I have high blood pressure or low blood pressure in the peritubular capillaries? Low blood pressure. And remember, why did I need to have low blood pressure within my peritubular capillaries? To allow a reabsorption to take place. So if I told you that angiotensin II is constricting my efferent arteriole. So what's going to happen to the pressure in my peritubular capillaries? Would I have a decline or an increase in the pressure of the blood in my peritubular capillaries? What do you think? If I'm constricting here, so you see, imagine on here, this is my efferent arteriole, and this is my capillary. I'm telling you that I'm performing the constriction here. So I'm narrowing down the diameter of the efferent arteriole. What do you think is going to happen to the pressure in the peritubular capillaries? Not in the efferent arteriole. I'm not asking about the pressure in here. I'm asking the pressure in my peritubular capillaries at this point. What do you think? So when you constrict the efferent arteriole, would you have more or less blood traveling into the peritubular capillaries? What do you think? More or less blood would be traveling in the peritubular capillaries. What do you think? Less or more? Less. Right, so if I'm constricting the efferent arteriole, less blood gonna be traveling to my peritubular capillaries. It's the continuation of the efferent artery. So if I have less blood traveling here, this means what in terms of the peritubular capillary pressure? What happens to the pressure in here as a result? Remember, if I have more blood, it's gonna create higher pressure. If I have less blood, what happens to the pressure? Lower pressure. So you see on here, I constricted the peritubular capillary, the efferent arteriole. And as I constrict the efferent arteriole, what happens to the pressure in my peritubular capillary? Decrease. And if I decrease the pressure within my peritubular capillaries, what's going to happen to the ability of the fluid to move back to the peritubular capillaries? If I have a decline in the pressure, remember the hydrostatic pressure is pushing fluid out. So I did decline the pushing force in the peritubular capillary. So my ability to allow the fluid to move back to my blood, did it increase or decrease when I reduced this pressure? So let me draw this on here. This is my peritubular capillary. And this is one of my kidney tubules, whatever, like proximal or distal convoluted tubule. If you remember, if you have fluid, it applies a pushing force called the hydrostatic pressure, as we've mentioned in the filtration. If I'm telling you that the source where I'm getting this blood into my peritubular capillary is closing, it's narrowed, it's constricted, and this is the effect of angiotensin II. You told me that less blood is going to be moving here. So I have a decline in the amount of blood in the peritubular capillaries. And if I have less blood, less fluid, would I be exerting the same pressure or less pressure? What do you think? If I'm pushing fluid back by a force of 10, now I have a decline in the amount of blood in the peritubular capillaries. So now the pressure will become 5 or will it become 15? What do you think? The pushing force. I reduce the amount of blood getting into the peritubular capillaries. So what happens to its pressure? The pressure drops. So it's going to become 5, lower than it used to. So this is what we're saying here. I constricted the efferent arteriole. This is the effect of angiotensin II. And as a result, the pressure dropped in the peritubular capillaries. As the pressure drops in the peritubular capillaries, my question now is your ability to get water back increase or decrease? So which one is going to be better, to move water back from the kidney tubule to your peritubular capillary if you did have a resistance of 10 or you've got a resistance of 5? Which one would it be better? Before you constrict the efferent arteriole or after you constrict the efferent arteriole? What do you think? So you see, I am moving here fluid due to the hydrostatic pressure in the tubule. Before I constrict, I did have a pressure of, let's say 10. I don't have 10 anymore, I have 5. So the resistance that is going to be pushing the fluid out has been reduced from 10 to 5. So would I be able to move more water back or less water back? If I reduce the resistance against the water movement, would you be able to move more or less water back? More water back. So as I move more water back, what happens to my blood volume? The amount of blood in my vessels will increase. And as a result, I've got an increase in my blood volume that results in an increase in the blood pressure. This is what I want. So angiotensin II, again, how does it increase the blood pressure? By constricting the efferent arteriole. The constriction of the efferent arteriole will cause a decrease in the peritubular capillary hydrostatic pressure. And as I reduce the peritubular capillary hydrostatic pressure, this will allow you to reabsorb much more fluid back to your vessels. And this results in an increase in blood volume, and so blood pressure. All right. Any questions? Any questions? Anybody is not convinced with any of the mechanisms of action of angiotensin II so far? So again, we've mentioned 4 main mechanisms by which angiotensin II is going to be increasing my blood pressure. First one is by constricting the arterioles all over the body. This will increase my total peripheral resistance and so my pressure. Another function is going to be by stimulating the reabsorption of sodium directly by acting directly on the kidney tubules and indirectly by stimulating the adrenal cortex to release aldosterone. Another mechanism of action is for the angiotensin II will be to stimulate the hypothalamic thirst center, which will drive you to drink a beverage, increasing your blood volume and so pressure, and stimulates the hypothalamus and a posterior pituitary gland to release ADH, which is going to be responsible to retain water. And as I retain water, this will increase my blood volume and so my blood pressure. Another functions that we just mentioned for angiotensin II would be to constrict the efferent arteriole. And when I constrict the efferent arteriole on here, this will reduce the amount of blood circulating in the peritubular capillaries, reducing the hydrostatic pressure. And as I reduce the hydrostatic pressure within my peritubular capillaries, the water will be able to move back to my blood from the kidney tubules. So this can enhance and increase my peritubular, my tubular reabsorption, my ability to get fluid back from the kidney tubules. One last mechanism of action for the angiotensin II is by enhancing the extraglomerular mesangial cells. You see on here, those are the extraglomerular mesangial cells to contract. And the contraction, try to think it this way. So let's say this is my extraglomerular mesangial cell. This is a very simplified way of thinking of it. This is my glomerulus. And this is my Bowman's capsule. So angiotensin II function here is to stimulate the extraglomerular mesangial cells to contract. So when I contract, what's going to happen? I'm reducing the size of the cell, and this is going to be pulling the capillaries away from the Bowman's capsule. Like this. So do you notice any difference on here? What was the area available for the filtration before I contract the extraglomerular mesangial cells? All this connection, all the parts where the vessel meets the Bowman's capsule. This is my surface area available for the filtration to take place. When I contract the extraglomerular mesangial cells, pulling the capillaries away, what happened to the surface area available for the filtration to take place? What do you think? Is it larger or smaller now after I contract the extraglomerular mesangial cells? It's much smaller, much smaller. So you see on here, I have less surface area available for filtration to take place. And as a result, what's going to happen to the amount of water that will be filtered out? Which one going to allow more fluid to get filtered? Before contraction or after contraction? Which one going to be losing much more fluid? 1 or 2? You're gonna lose more fluid. One. So what happens? What happens to the amount of water that I lose out in the kidney tubules after the contraction of the extraglomerular mesangial cells? The water loss is going to be reduced. And if I have a reduction in the amount of water that I am losing out, what's going to happen to my blood volume? If you're not losing water out in the urine, you're gaining blood volume. And if your blood volume goes up, what's going to happen to your blood pressure as a result? Your blood pressure increases. All right. Any problem with those concepts that we've just discussed? If I would pick just one topic as the single most important topic in those two chapters, the urinary system and body fluids, is going to be this topic: juxtaglomerular apparatus, how it functions, its structure, and the actions of angiotensin II by which it increases the blood pressure. This is going to be the single most important topic in those two chapters, and actually it's half of the essay question. So the essay questions that you're going to have in this upcoming lab exam, it's going to be addressing how do you regulate the blood pressure. So regulation of the blood pressure, if I have a low blood pressure, this is what I will do. And we're going to discuss how do I respond in case of high blood pressure later in the next sessions. All right, so what happens if I have a drop in my blood pressure? Drop in the blood pressure, this will be detected by the granular cells of juxtaglomerular apparatus. And the granular cells will be detecting the drop in the blood pressure by detecting the reduction in the degree of stretch of the afferent arteriole. This will stimulate the cells to release renin. Renin is going to be converting angiotensinogen into angiotensin I that gets converted by angiotensin converting enzyme, which is present in the endothelium of the capillaries, into angiotensin II, which is the active form of the hormone. Angiotensin II gonna be increasing the blood pressure by multiple means. First one, constriction of the arterioles, which will cause an increase in the total peripheral resistance and so the blood pressure. It's also gonna be enhancing the sodium reabsorption directly by acting on the kidney tubules, and indirectly by stimulating the adrenal cortex to release aldosterone. Third mechanism of action of angiotensin II will be to stimulate the hypothalamic thirst center. This drives me to drink a beverage and so increasing the blood volume and pressure. And the stimulation of the neurons responsible for the formation and the release of ADH, antidiuretic hormone. Antidiuretic hormone acts by retaining water from the collecting ducts by inserting aquaporine channels in the tubular cells, which will allow you to retain water, increasing the blood volume and so the blood pressure. Another actions that we've seen for angiotensin II was to constrict the efferent arteriole, and as I constrict the efferent arterioles, this will cause a drop in the hydrostatic pressure of the peritubular capillaries, allowing the fluid reabsorption to my blood, and as a result, an increase in blood volume resulting in increase in the blood pressure. Another mechanism of action for angiotensin II will be to stimulate the extraglomerular mesangial cells to contract, and as I contract the extraglomerular mesangial cells, this will reduce the surface area available for filtration. As less water gets lost out in the filtrate, this means this water gonna be retained in my blood vessels, increasing my blood volume, and so my blood pressure. Any questions? Any questions? All right, so we can have a 10 minutes break. We come back and discuss more about the blood pressure regulation and the urine formation.